Okay, so welcome to all the participants on the part two of a webinar on augmented reality microscopy, ARM. We have with me Dr. Dan Rudmann, the lead for digital pathology at Charles River Laboratories, and Dr. Liron Pantanowitz. He's the director of anatomic pathology at the University of Michigan. And again, my name is Gabe Siegel. I'm the CEO of Augmentiqs, a company that develops ARM technology. So this is the second part of a two-part series on ARM microscopy, and following the first part, the first webinar, which was pre-recorded. So there were a number of questions which came in from individuals, and what we wanted to do is to address these questions. So the first part will be a Q&A, and I guess the way that it'll work is each question we'll address it to Dr. Pantanowitz and Dr. Rudmann, and they'll each give an answer, and they could feel open discussion on their thoughts on the questions. And of course, if anyone in the audience wants to join in, wants to add additional questions to it or additional thoughts, we'll try to figure out a way to make that happen. After that, we could again at the end we'll open it up to additional questions, and then we'll have like just a sort of a conclusion for the thoughts, future predictions for what ARM could do for digital pathology. So so let's get started. The first question. The question is written here. I think that maybe it'll be best to maybe I should read it in case there are some people which are watching that aren't watching on a computer screen. So the question is like this. I work for a company that develops AI algorithms. And until this point, we've focused on developing algorithms for WSI. So the concept of deploying AI algorithms within the microscope is interesting and appears technically easy to accomplish. However, I wish to hear your input on the actual need for such a solution and what algorithm would, in your opinion, would be most advantageous to the pathologist for an AI company to develop. So that's the first part of the question. Maybe I'll stop there. And or do you think that we should just say the entire question?
Yeah, read the whole question and we can answer them.
Okay, so as a second question, I also wish to mention several points that I believe WSI has an advantage over ARM, and I'd like to receive your input. The use of AI on a WSI file has advantages over the glass slide, and that an algorithm can perform the analysis as soon as the slide is digitized. With ARM, it appears that the AI analysis can only be performed when the slide is on the microscope and pathologist is working. ARM seems to reduce the actual need for AI, as in the majority of instances, the pathologist doesn't require AI, whereas with WSI, artificial intelligence can be used to pre-screen and prioritize the digitized slide prior to their being viewed by a pathologist, which is, in my opinion, a game changer of using AI. Furthermore, performing AI on a WSI file means that the algorithm takes into account the entire sample, whereas ARM is only the field of view. Would you say that the quantitative results of the WSI file would therefore be more accurate than just a field of view? Knowing that more accurate doesn't necessarily result in a different diagnosis. And finally, there are certain pathology samples where a field of view is not enough to render a diagnosis. Prostate, for example, where we run algorithms on the entire WSI file. What is your opinion on these tissue samples like prostate? Liron, would you like to start?
Yeah, all right, thanks, Gabe. So I have three separate answers to that multi-part question. The first thing is, when you're making AI for WSI, you cannot use the AI unless you have WSI. So the truth is, most pathologists in the world have a microscope, but very few have WSI. So if you're making AI for WSI, you're making it for probably less than 1% of pathologists around the world. And while one day we may get there, this could be 5, 10 years, what are we going to do in the interim? So that's the first thing I would say. The second thing is, if you're making AI for whole slide imaging, there's no reason you cannot apply the same AI to augmented reality microscopy. What this person asking the question points out, well, there are advantages to the WSI. And I would say that actually one of the problems today with people making AI algorithms for whole slide imaging is they are making AI algorithms to replace pathologists and not augment pathologists, which is a huge problem. And I think that with ARM coupled with AI, probably the best solution we have now. For example, if I have a case of breast cancer that has extensive DCIS and only a small area of microinvasion, I'll bet that there are very few AI algorithms now that will get that right every single time. Exclude all the DCIS, diagnose that and grade that, and then find the small area of microinvasion amongst that, and then separately grade that. So if you're going to try and implement a system to accomplish that, you need a highly sophisticated AI algorithm, and I doubt you would get 100% performance on that. However, if you add the pathologist to that equation, let the pathologist go find you the invasion and then deploy the AI. What's the grade of the DCIS? What's the grade of the the invasive cancer component? So there's no reason why you cannot couple the pathologist with AI instead of have the AI replace pathologists. And the third thing is, if people building AI algorithms today for WSI, the main problem with that is that we're talking about very narrow AI. All they do is one task, and I'm afraid that they don't really understand what we do as pathologists. Neither what I do or Dan does. There's much more that goes into interpreting a case and then reporting that out than an AI algorithm can currently do today. We are not at the point where we have super intelligent AI. These AI algorithms that are being built are very narrow in that they could just do one thing. So for example, when I look at prostate cancer, there are AI algorithms that are quite good at now detecting prostate cancer, diagnosing cancer, grading it, and so forth. But what I do is, in addition to that, I'm ready for any rare variant that comes along my way. I'm also ready to diagnose not just adenocarcinoma, but other features in that case. What if there's background granulomas or something else that's important? So that's why I think AI algorithms for WSI, while they're great, there's a long way for them to go before they replace us. So I think ARM has a great role for coupling the AI with the pathologist and not eliminating us for now.
Yeah, so totally in line, and it's wonderful how the preclinical space really just really aligns everything that was just mentioned. So for sure, I mean, while we have whole slide imaging capabilities at some larger facilities, easy access for people to be able to tap into those, I'll be honest that in toxicologic pathology, 99% is still the glass slide. And even when whole slide imaging becomes more part of our industry as kind of the staple for our primary evaluation, there's still going to be a lot of consultants, a lot of smaller groups that are just not going to probably do WSI just because of the logistics of cost and complexity. And so that's where ARM fits in. So I totally agree. It's the same picture in non-clinical pathology as it sounds like it is for clinical pathology, which doesn't surprise me. I also totally agree with the synergism, because so think about it when you're driving one of those fancy Tesla cars that does the auto drive for you. Okay, so if you're sitting back and letting the car do everything, is that better than you interacting with the car in a synergistic way to produce a better outcome? And that is a safe drive where you get there efficiently. I think it's the synergism, because you've heard, unfortunately, of the stories where people are falling asleep while they're letting the car drive themselves, and then it has a terrible accident. So that's what we don't want to do as pathologists, right? So it's the synergism. And when you do the whole slide imaging in advance, and then you provide it to the pathologist, if you can do that, then the synergism is not quite as much there as when you're actually you're looking at the pattern, you're doing the pattern analysis real time, and then you're incorporating the AI as you're looking through the microscope. As your mind is thinking, it's almost like having another pathologist on a multi-headed scope with you at the same time, but instead of another pathologist, it's a computer. And that's the base, and the basis of that computer is the ground truth of pathologists. right? So it's really a very interesting approach. And so I think that it's all about intended use. I think there's a place for whole slide imaging. There's a place for AI and whole slide imaging. There's a place for where that can bring extremely good value to both of our industries. But there's also a place for ARM and its considerations. And again, it's all about really how you want to use it and apply it. And let's see. I think that covers field of view for us in preclinical toxicology. We look at a whole tissue section. So we aren't clinical pathology generally. Much of it is biopsy evaluation for good reason. We in non-clinical have access to the whole organ, which is a benefit for us in some ways, but it's a lot more tissue to look at. So, and to be honest with you, I think one of the things that we deal with in non-clinical toxicology, it maybe our clinical colleagues in human medicine don't have to deal with as much as a lot of what we look at is normal. Because we are looking at a large tissue section, right? We're not looking at a biopsy coming out of a mass or something like that. So I think for us, maybe as whole slide imaging gets more incorporated, there may be some more advantages for a whole slide imaging AI approach for us for screening than maybe it would be in the clinical world. So that may be where there's a little bit of a difference in the non-clinical and the clinical space. I think ARM is equally intriguing and helpful for both spaces, but possibly whole slide imaging AI screening approaches in a non-clinical environment where we're trying to avoid false negatives and kind of just alert the pathologist to abnormalities might be a little bit more advantageous for us than the clinical colleagues. But I'd be interesting to hear what Liron thinks about that. But that's maybe the only difference, I would say.
Yeah. So Gabe, I think we're aligned. Doesn't matter what field of pathology. It's still pathology. So yeah, we see equal benefit in ARM. Very
interesting. So we'll move on to the next question. Again, if anyone wants to, if any of the participants wishes to add something, I think there's like a chat box or some type of hand raising tool. Let's see. Okay, so here we are. I understood from the webinar that ARM is not necessarily an expensive technology, yet unlike WSI, which our lab has acquired, an ARM would be required for each microscope, while WSI can be a single system. What I'm interested in hearing is how in your labs you determine the cost benefit of ARM.
I can start with this one if you want me to, just since you took the last one on, and then I think it's a good question if you want me to, Gabe.
Go ahead.
Just to share the person that takes the first step forward. Yeah. So it's going to be partly dependent on the structure of the organization. So, if you have a consultant practice, right, where you're the only pathologist, buying a whole slide scanner for yourself, it's just not a feasible. I mean, likely not a feasible economic return on investment. So yeah, if you can access a system that works with your microscope that you've already invested in, right? That can use ARM, and then apply tools and algorithms that have been developed, then that's going to be much more cost effective for that kind of single pathologist practice. And in toxicologic pathology, there is a huge area. I mean, there's a large number of pathologists that work as single consultant entities out there, and to give them the opportunity to take advantage of ARM to help them in their practice would be very very useful. Now, for my company, like Charles River, I mean, we have over 150 pathologists across the globe, multiple sites. So clearly for us, putting a single unit on all 160 microscopes. I mean, we could do it, but yeah, we could also take advantage of a network of scanners that can interact together. So then the return on investment changes a bit. But again, I'll tell you right now that we are an unusual company. There's not a lot of companies that have 150 pathologists. In fact, we're probably the only company. Most companies are going to be, a few pathologists, and maybe a dozen pathologists at the larger end. So again, I think it's intended use. It's kind of based on the structure of your company, based on your needs too. I think, for instance, we looked at over 4 million tissues last year. So is it realistic to scan all 4 million tissues when really you only want to do image analysis and maybe AI on 10% of those? I mean, is that worth the extra, all that time and money, right? Or do you want to make it more arm focused where the pathologist chooses what to apply the arm to? And so you save all that overhead, right, of scanning and everything, and the slide comes right out of the lab, it goes on the microscope with the pathologist, and the pathologist has a toolkit of AI algorithms that he or she can apply on the microscope. So again, even in a large company that has potential to scan a lot, do you need to scan it all to add value? So again, these are the types of discussions and things that people need to think about. It's not a simple answer, but I hopefully gave you some perspectives on how it could be thought about differently with ROI.
Yeah, Gabe, so my perspective on this question is as follows. Many people underestimate how expensive it is to implement whole slide imaging because they think WSI is the scanner. In fact, the scanner is the cheapest part of the whole thing. When you talk about, as the question actually says, the WSI system, there's the scanner, then you need the image and case management software and a license and perpetual maintenance on that. Then you need a server to at least host the images, and if you want to keep them for longer, that's even more expensive. You need a digital cockpit for the person to work at where the viewer will work, okay? And if you want an FDA cleared system here in the US, then those are quite pricey. And guess what? The slides don't scan themselves. So you have to actually pay an FTE person to scan that, pay their benefits every year. When you scale that up, it's more FTEs, and you need someone to manage all of that. So whether you're paying a central IT or someone to manage that, actually a WSI system is very expensive. To get started, we're talking millions of dollars up front, and then to maintain that operation. Whereas today, every pathologist in the world has a microscope. If you're an anatomical pathologist in one form or another, even a microbiologist, you already have a microscope. So that's already done. And, not every pathologist in the world has a WSI system. In fact, I think it's going to be more than a decade before everyone gets a WSI system. I'm talking in developing countries too. Today, I think I could walk into any country in Africa where there's a pathologist practicing, and they wouldn't be practicing if they didn't have a microscope. So they already have a microscope. So, I think when we're talking about bringing the power and flexibility of digital imaging to every pathologist, the augmented reality solution now is a much more cost-effective solution to bring this technology, including AI, to everyone today. All they need to do is buy a system and get a computer to do that. So that's what I think. I think people underestimate WSI, and unfortunately, if you're saying you want to do AI with WSI, you cannot do AI without the WSI. So you actually have to outlay several millions of dollars, and then only can you realize the benefit of AI. And this is a great workaround that solves that problem now. If you just, as Dan said, if you just don't have the money, if you're even a single person, there's no a return on investment for millions of dollars just to start using AI.
Yeah. And I think the other great thing about ARM is, okay, so us pathologists, we're old souls, I mean, I don't know if you guys can see my microscope next to me. You know, I'm a digital pathology guy, but guess what? My microscope's sitting right next to me. It's like a stuffed animal. You know, I love it. And to go, you know, for us to expect all pathologists around the world to go from what we've been trained on and what we're used to using, we like to feel with a microscope and the focus rings and all that, to go that to all WSI and AI and be effective and efficient and even just accept it is probably, that's a pretty big objective. But ARM is cool because you still have the light microscope, but again, it's like it's like a dual-headed scope in a way, but with a computer, and so it's kind of a nice transition and we can learn a lot from it too, right? So the other thing about WSI AI is, you know, you're going to have a bunch of trainers that pathologists are going to be working with computer scientists to make these AI algorithms using WSI. And then you're going to be giving these tools to pathologists that weren't involved. Well, so when you're using the microscope and using ARM, you feel like you're involved, right? A little bit more. And so there's a little bit more, I guess, interaction for the pathologist, not only using their scope, but also in terms of tweaking how they need to use the AI to help them. And I think that's got some value that's different than the value you get from a WSI approach. So again, I don't think they're conflicting with each other. There are some limitations based on financials and other things for sure, and I totally agree that it is a huge infrastructure to build to do it right for WSI. But I think they're complementary, and I think that the ARM is possibly for a lot of pathologists a great transition into digital pathology.
Yeah, Dan, I'd like to add that, you know, and you know this probably as well as any other pathologist, not every slide in the world is going to be able to be digitized, right? There are some that you just cannot focus on. They're old slides, they're too thick, they're broken, someone patched them up. There's polarizable material on there that you just cannot, you know, handle with digital imaging today. Cytology material people complain about. You may need to put oil on a slide to look at it. So, you know, what happens to all of those slides, which there are millions of those being looked at around the world? Are we just going to eliminate those on all those patients, or, you know, veterinary patients that you have? Do we just exclude them from all the benefits? So that makes no sense, right? So we need a solution for those cases that you just cannot digitize. And you know, I think ARM fills that gap. The current technology we have on the market cannot address that for now.
Yeah, I totally agree.
Okay, so let's move on to the next one. So time is flying by. According to the 2018 Toxicologic Pathology study brought in the webinar, ARM can be used for real-time telepathology for peer review. Yet it was also mentioned in the webinar that there is no guideline for validation and licensing. Can you please provide more details on this point? Do you believe ARM or any other microscope camera with some screen sharing application can be used for telepathology? I'm asking specifically for GLP studies and peer review, though I imagine there are similar requirements for clinical pathology. And as a second question, there is value in having a WSI file for telepathology enables me to view the entire slide. I believe there may be reduced value of the ARM approach where I can't see the entire slide. So this is, Dan, this is also probably a good question for you to start with.
Sure. Yeah, so, you know, the best practice of peer review in non-clinical pathology and toxicology is not a raw, it doesn't produce data. So it's basically a consultation exercise where there's a second pathologist that is interacting with the study pathologist, who is responsible for the data and who is responsible for the final diagnostic decisions and writing the report. So really anything that we use in the peer reviewer uses to help them in decision support to help them make a decision whether they agree or disagree with the pathologist is again, just what it is. It's decision support. It's not producing data. It's really just helping them make decisions. And so it's a different type of regulatory requirement. I mean, if the peer review pathologist was trying, let me use an example, the opposite example, wanted to quantify a specific finding, let's just say mitotic index, and wanted to report out a number using ARM of a mitotic index for a series of slides, that's different because it's producing data. But if you're just using a system to help you make a decision, then it's a different situation. So I think that the regulatory issues are different. Now, that said, we obviously want to qualify the systems. We want to make sure these systems work well and that they meet specific requirements, so that, because we don't want to give decision support tools to pathologists that aren't useful. And let me just give you a really crude example. If I'm hitting the literature and I'm reading and I'm trying to use the literature to help me make decisions, if I look at, I might avoid certain journals because I know, be honest with you, that the journals, the articles that come out there, I don't trust them. I don't feel like they're going to provide good quality decision support material for me. But I'll go to other journals where I know that I can rely on that information to help me make decisions. It's the same thing, right? We want the pathologists to make sure that they can rely on these tools that we're building for them, whether they're, they be AI-driven tools or just literature. And so that's important. So the qualification will be very solid, but the GLP requirements are going to be different because it's not producing raw data. That's for peer review specifically. Now, as a second question, I mean, you can look at the whole slide with ARM. I mean, you've got the slide underneath the microscope and you move it around, right? So now you can't, it's not like you're going to get one image right away, right? Because you're going to only be able to look at your field of view. But that's really the powerful thing about it is, right? Because the pathologist will help get to the area of interest through their training and experience where they want to use the ARM to help them make a decision. So instead of, let's say if you have a whole slide image and let's say 95% of it, who cares what the artificial intelligence thinks about, because it's just normal. And there's only 5% of that huge file that really is of interest that would really help the pathologist make a decision. So someone could argue that other 95% is just wasted time and energy, right? I mean, why waste your time scanning that? Why waste your time building the infrastructure and scan that? Well, if you have a slide underneath the microscope and you have a trained pathologist that looks at the slide and says, I'm really interested to see what the computer thinks about this part of the slide. He or she goes right to that part of the slide, boom, gets to it and gets the decision support he or she needs. So I think it's a key difference to think about. I understand people always think more data is better, but sometimes more data is not better. Sometimes more data is more time, less efficiency, and distracting. So anyway, my thoughts.
Yeah, a similar thing, Gabe, to clinical practice is if you need to use telepathology to support your practice, you need to pick the right technology to fit that and not overfit that with expensive, unnecessary technology. So if you have a situation where you do not have a skilled person on site and someone's just loading a slide in a box and they know nothing about pathology, fine. Then you need access to the whole slide and control of that, whether it's a whole slide imaging system or a robotic system that does telemicroscopy and you can navigate. Those are expensive to buy and expensive to maintain. But if you have a skilled person on site already, like another pathologist or a technologist, then to buy a whole slide imaging system or a whole robotic system is an overkill and it's unnecessary, kind of is what Dan said. Then all you need is, because that skilled person on site is actually doing the heavy lifting, getting to the right area, but they just need some support, second opinion right there and then, or advice on how to proceed. And so, ARM is then a perfect solution for that. You don't have to overfit the technology to address that specific need. So I think, it's what you need. This provides a simple solution for that, not overly expensive and a waste of a lot of technology to maintain.
Okay, we'll move on. That was a great answer. We'll move on to the next one. Can you provide more details about how you see this ARM technology being used for pathology education? In the school I teach at, we rely heavily on WSI, and I imagine this is the case for most medical schools in the country. By the way, is this the case for the University of Michigan?
Yeah, so most, Dan, I'll answer this and then you can, follow, from veterinary practice. Most medical schools and actually allied health schools, dental, etc., have gotten rid of microscopes. They're expensive to maintain. Even trying to maintain the slides that people look at are expensive, and people don't include even the rare cases because it's hard to find a rare case, and you certainly get a lot of recuts. So digital pathology has really been great from that point of view, whole slide imaging. Can ARM provide similar, functionality for education? I actually haven't thought about that because I haven't used it yet, but it definitely can. So 2 examples would be, number one, if you are at a medical school where you could not invest in whole slide imaging technology, or even if you have it, you could still have your teacher up front with your learners, and you could broadcast then just from one microscope so everyone could see what's going on. If you don't have, someone who could teach you, at least then if you have a microscope, many schools around the world have microscopes still, you could introduce some self-tutoring, AI or etc. So in other words, what you could do is you could look at the slide, but if the person cannot find the plasma cells or the eosinophils, then you turn on real-time annotation and it points out, look, yeah, the eosinophils. Oh, fantastic. So you can help learners with microscopes wherever they are. So, I haven't done this personally, but I certainly see that, augmented reality microscopy can do that to assist with education.
Yeah, I agree. I think both can. I think our veterinary schools and training programs are a little bit farther behind than the medical practices, mostly just because of funding and size. And so there's probably a little bit more dependency on the light microscope than whole slide imaging, although whole slide imaging definitely is being used for seminar type of learning and databases and those types of things and training. So I think both could be very beneficial. And again, I think the most important thing for us, for people like us that are really interested in digital pathology, is to help transition our colleagues into it, because it is a big change for people, and I think ARM can be kind of a nice transition, and still have its own utility within its own space. So.
Technology seems pretty straightforward. ARM is a camera, projector, and a stage camera. Assuming the projector and stage camera are not used, is ARM any better than a normal microscope camera?
All right, I'm happy to give you my opinion. Augmented reality microscopy is a camera with added value. If you're going to buy it, why wouldn't you want to? It's like I would tell someone is, why go buy a car with a turbo? If you're never going to turn on the turbo, then don't go buy the car with the turbo. But if you buy the car with a turbo, then you get the benefit of driving with a turbo, which is way more fun than it is just to drive it without that. So that's what I would say. You're buying a turbo engine microscope. So, turn on the turbo and go for a ride.
Yeah, exactly. Just basic cameras don't have the ability to integrate AI and machine learning and everything in it. So that's really the difference, right? So, it's basically just a telepathology tool, or just a tool to take pictures for communication and presentations and things. But an ARM system will have the benefits of both. Basically the computer assisted machine learning type of technology, along with the digital camera technology. So it's really you're just getting more, as was said.
So in terms of reporting requirements for regulatory submissions, obviously more from the point of view of the CRO than a regulatory body, do you believe there is any potential for ARM, in particular data such as histopathology images, slide label, position of image on the slide? What are your thoughts on including it with the rest of the documentation? And do you believe there are other aspects of regulatory submission that ARM could be beneficial to? So this is definitely, Dan, addressed to you.
Yeah, it's all about intended use. So again, if you're using ARM as a way to produce data, so basically a quantitation, maybe a quantification, or specifically, again, I think a great, if you look at just some of the papers that have been published on, measuring Ki-67, for instance. So, let's say you're in a preclinical study and you want to measure Ki-67, and we want to quantify it, but yet maybe historically it's been more of a semi-quantitative method that a pathologist use, or maybe they just were counting, on a microscope slide. And then and let's say you use ARM for that, right? Instead, right? And it quantifies that key 67 readout, and then you include that in a pathology report. That would be producing, that would be a raw data source for that pathology report. So that would have to undergo, if it's part of a GLP study, good laboratory practice study, it would have to go undergo validation. But you know what? I'll be honest with you. We've been validating machine learning for decades. I mean, I've been at several companies where we've developed machine learning algorithms that we've used to as part of submissions to regulatory agencies. There's well-defined computer validation methodologies by the regulatory agencies. There's a lot of people out there that know how to validate systems. So it's really not it's not a barrier. It's just a matter of, do we need to validate it or not? That's the decision. There's no reason to validate if you don't need to. If it's decision support, if it's just like accessing the literature, accessing another pathologist, and using a computer just to help you make decisions, that's one thing. If you're using to produce raw data, that's going to be the basis for a pathology report, that's another thing.
Okay.
Yeah, and
Liron, do you want to add anything?
I'm not an expert in this area, but I don't see why you couldn't track, right? You track the barcode, you track the GPS equivalent coordinates as you're looking at it real time. So it's not that all of a sudden now you cannot track anything. Maybe not to the great digital extent, but there's no reason why you couldn't track slides, barcodes, GPS location, and so forth.
Excellent. Okay, so that was all the questions. Let's see. The whole presentation, it was jumping. Now it doesn't want to move on. So if any of the participants, I guess this is the opportunity to send in something by chat, but if nothing comes in, I guess concluding thoughts and future predictions. Liron, do you want to start?
Yeah. So, I was excited to see this introduction of augmented reality microscopy introduced. I was happy to, test it out for several projects. I'm still working on projects, exploring the use of this. My recommendation would be for pathologists out there, if you have a microscope, slap on a turbo engine and give it a test drive. You'll be amazed at what you can do. You do not have to overcome that inertia of trying to get a slide digitized first if you don't have that system, and so forth. I think it's a great opportunity for us to capitalize on what's happening now, whether it's just simple annotations, which we're doing now. I'm working with my fellow Mustafa Yusuf. We're just doing simple annotations, which is so much easier in real time versus, being able to run image analysis. And, I've done those Ki-67 studies. So it opens up the world for you on your microscope. And as I've mentioned earlier, don't have to first digitize the slide, even those difficult cytology slides or whatever that I have that I cannot easily digitize today. So, I can get going right away with my turbocharged microscope.
Yeah, I agree. It's a great way to, automatically the system provides just the non-turbo charged benefits, which is just the ability to collaborate, the ability to interact, the ability to capture images, all this stuff that we can just do with basic cameras. But then again, it gives you an entry point, which is a very reasonable entry point to start experimenting with machine learning, annotations, and things, which again, I think really is going to be the future for us in pathology. I think, I do not see machine learning, AI-driven machine learning at all taking our positions. I kind of look at it this way. I think the pathologists that kind of adopt AI machine learning as a tool in their arsenal to be more impactful on medical science are the pathologists that are going to really succeed in the future. And so why not start now? Why not, get involved with these types of systems, which are, again, a lot less expensive than a whole slide imaging system, and that are part of your microscope, which is great because you're used to using your microscope, and then experiment and start. Let's build together the future of AI ML and its impact on both clinical and non-clinical pathology. It's an exciting time for us. I mean, I think this decade is going to be the most exciting ever for pathology in terms of its interaction with the computer. Let's make it happen. The more people that get involved, the better. And I know a lot of people can't get involved if they depend on WSI. So I think ARM is a great entry point. Don't do it though. Don't buy it just to buy it. I mean, the worst thing you want to do is buy, i7 computer when you only need an i3, right? So again, if you make the investment, get involved and apply machine learning, and people like, groups like Gabe's are very excited about partnering with us. And there's a lot of other companies out there interested in partnering with us. So, and internal people that are computer scientists that are interested in partnering with you. So it's a great way also for us to build relationships with computer engineers, computer scientists, because if we let them do their own thing and we do our own thing, then unfortunately our solutions probably aren't going to be the greatest. If we work together, our solutions are going to be fantastic.
So a little over what we thought it would take, but I was fascinated. I thought it was really, really interesting. So thank you both so much for taking your time to take part of this webinar. And available for everyone. And sorry, not live, make it a recording available. So great. So thanks so much and have a great day.
Bye, everyone.
Thank you. Bye now.